So I'm in AP Biology this year, so I've decided to make a series of videos to help review for the AP Bio exam. So this video will be about the first unit, Biochemistry. Okay, so let's start with the atom. The atom has several parts: protons, which are positively charged, electrons, which are negatively charged, neutrons, which have no charge, and the nucleus is the center of the atom and is made up of neutrons and protons. And usually the number of protons is equal to the number of electrons, so there's a neutral charge. However, there are also these things called isotopes, which differ in the number of neutrons. I just think it's important to have a basic understanding of the atom in order to do more biology. So here on the right, you can see a picture of an atom with the nucleus in the center, protons, neutrons, and electrons orbiting around. So we have excited and ground states. The ground state is when all the electrons are in the lowest energy levels available. And then there's the excited state, where the electron moves to a higher energy level when it absorbs energy. And it releases energy when it returns to the ground state. So as you can see in the little diagram that I drew, when it goes from a lower energy level to a higher energy level, energy is absorbed. And when it goes back to a lower energy level, energy is released. So now let's talk about chemical bonds, starting with covalent bonds. So covalent bonds are the sharing of electrons. And now let's talk about electronegativity. So that's the attraction of one of the atoms for the electrons in the covalent bond. And this leads to polar covalent bonds, where the electrons are shared unequally between atoms. And again, this is due to a difference in electronegativity between the atoms. An example is water. And this leads to partial positive and negative charges in the molecule. So this is a picture of a diagram of water. And as you can see, there's a partial negative charge around the oxygen atom, which is the larger atom, and partial positive charges along the two hydrogen atoms. And that's because oxygen is much more electronegative than hydrogen, so the electrons will orbit around oxygen more, giving it a weak negative charge. And again, this is a polar molecule, which means that the electrons are not shared equally, and this will be really important as we move on. And there's also nonpolar covalent bonds, so that's the opposite, right? Electrons are being shared equally between the atoms. An example is oxygen gas. As you can see in the picture, there's no partial negatives, no partial positives, because the electrons are shared equally between the two oxygen atoms. And that's a nonpolar molecule. And this polarity brings us to hydrophobic and hydrophilic substances. So we have hydrophilic substances first, which are water-loving. That's what hydrophilic means. And these are polar molecules, right? Because we talked about how water is polar. And water being polar makes it attracted to other polar molecules that have unequal sharing of electrons. And hydrophilic substances could be charged or ionic. And all of these hydrophilic substances will dissolve in water, which is why water is called the universal solvent, because so many molecules will dissolve in it. And again, this is all because of the polar form of water. Because water is polar, it is attracted to other polar charged and ionic substances. And then there are hydrophobic substances, which are the opposite. They are water-hating. And these are nonpolar substances. An example is oil. And it's important to note that like dissolves like. So hydropolar molecules will dissolve in a polar substance, and hydrophobic substances will dissolve in other hydrophobic substances. So as you can see in the picture, that's vinegar and oil. The two don't mix. Vinegar is hydrophilic, while the oil is hydrophobic, so they won't dissolve in each other and they'll stay separate. Next we have ionic bonds. So in this type of bond, one atom, quote unquote, steals electrons from the other. And this is because the difference in electronegativity is so large that the more electronegative atom completely takes away an electron from the other. And it becomes an ion, which is a charged atom or molecule. And then we have cations, which is the positively charged ion. So that's the atom that will lose the electron. And we have anions, which are negatively charged ions, and that's the atom that gained the extra electrons. And these two will be together in an ionic bond, and that's the attraction between cations and anions. So if we look at this example, sorry, a little bit of the names got cut off, but we start with a sodium atom and a chlorine atom. And if we look, sodium has one single electron on its outermost valence level, and chlorine is missing one electron on its outermost valence level to have a complete outer shell. So, as you can see with the arrow, chlorine will completely take an electron away from sodium. So now sodium has a completely full outer level, and chlorine will now have a full valence level. So sodium loses an electron, so it becomes a cation, right? It has a positive charge of 1+, and chlorine will gain another electron, giving it a negative charge, so it's the anion, and it'll have a charge of -1. And these two ions will stay together in an ionic bond. So now let's talk about hydrogen bonds, which are very important. This is the attraction between a hydrogen atom that's in a covalent bond with an electronegative atom and another electronegative atom. So this is what keeps water molecules together and is what leads to many of water's unique properties. So if we look at the picture, there's a hydrogen bond between one of the hydrogens of a water molecule to the oxygen of another water molecule. So it's important to note that in a water molecule itself, the hydrogens are not connected to the oxygen by hydrogen bonds. They're connected by covalent bonds. But between water molecules, there are hydrogen bonds. So now let's talk about the properties of water. It has many unique properties. Let's start with cohesion. So cohesion is sticking to itself, basically. So hydrogen bonds, like we talked about, hold water together, and you can see that in the image with the little molecules. There's also high surface tension, which means that water's surface is more difficult to break through than most other liquids. And again, this is because of the cohesion of the water. And as you can see in the picture of the paperclip, the surface of the water is kind of tough to break through, so the paperclip can sit on top without falling through. And we also have adhesion, which is basically water sticking to other substances. And if you look at the picture of the stem, yes, it's a little complicated, there's a lot going on, but let's just focus on the adhesion and cohesion, and you can see that water molecules' adhesive properties with the sides of the stem and cohesion with other water molecules allows water to go up a stem of a plant, which is pretty neat. So now let's talk about moderation of temperature. So water has high specific heat. So what is specific heat? Specific heat is the amount of energy needed to raise the temperature of 1 gram of a substance by 1 degree Celsius, which means that water resists large changes in temperature because it has a high specific heat. And stable ocean temperatures are really important because it provides a good environment for marine life. Here's a nice picture of some marine life. And water also has a high heat of vaporization. So heat of vaporization is the amount of energy needed to convert 1 gram of liquid to a gas. So again, this helps moderate the climate. A lot of heat from the sun is absorbed by seas, and the good thing is that a lot of this heat is used in the evaporation process. So again, this is all about water being stable and having a stable temperature, which is really important for life. And it also has evaporative cooling. So basically, evaporative cooling means that the hottest molecules evaporate first, right? Leaving behind the colder ones. So again, this is contributing to the stability of temperature. And an example of this is sweating, right? When you've done, let's say, exercise and you're really hot, your body sweats, and that sweating will cool you down. Another neat thing about water is ice. Ice is less dense than water, which is not always the case for liquids and solids. A lot of times the solid is more dense than the liquid, but in this case, the solid is less dense, which means that ice will float in water, and this insulates the liquid water below it. So this will protect all bodies of water from freezing completely because the top layer will become ice, and will float above the water, and the water won't freeze. So here there's a picture of the molecules in ice, and it's less dense than water because of the way the molecules crystallize and stay far apart in ice. And below there's a nice picture of a frozen lake. And again, this is really important because it saves all the life below the ice. Now let's talk about pH. pH measures the concentration of hydrogen ions in moles per liter. And this formula for pH is negative log of the concentration of hydrogen ions. All the brackets mean is concentration. So in a solution at 25 degrees Celsius, the concentration of hydrogen ions times the concentration of hydroxide ions is equal to 10 to the negative 14th. And the pH of pure water is 7, which means that the concentration of hydrogen ions is equal to that of hydroxide ions. If the concentration of hydrogen ions is greater than the concentration of hydroxide ions, then the solution is acidic, which means the pH is less than 7. If the concentration of hydrogen ions is less than the concentration of hydroxide ions, the solution is basic, which means the pH is greater than 7. So on the right, you can see a nice little scale of what what the pH is of different substances. So now let's do an example, because all of that was a little confusing. So let's say that at 25 degrees Celsius, a solution has a hydroxide concentration of 10 to the negative 5th. What is the concentration of hydrogen ions? What is the pH? And is the solution an acid or base? So let's start by writing down what we know. We know that the concentration of hydroxide ions is 10 to the negative 5th. We know that our little formula for solutions at 25 degrees Celsius is that the concentration of hydrogen ions times the concentration of hydroxide ions is equal to 10 to the negative 14th. And we know that to find the pH of something, we do negative log of the hydrogen ion concentration. So let's do step 2, which is finding the concentration of hydrogen ions. So we can use our little formula from above and substitute the concentration of hydroxide ions with 10 to the negative 5th. And if we divide by 10 to the negative 5th on both sides, again, remember your rules for dividing with exponents, we will get that the hydrogen ion concentration is 10 to the negative 9th. Now let's find the pH. And again, use our formula and plug in 10 to the negative 9th for concentration of hydrogen ions. And for those of you that don't know what log is, for now, log in this case is base 10. So what that means is that we're just taking the exponent of 10 to the negative 9, because it's log base 10. So negative times negative 9 will give us positive 9, so the pH is 9. And now let's determine if it's an acid or a base. So we don't know the relationship between these two, but we'll figure it out. And now I plugged in 10 to the negative 9th for the concentration of hydrogen ions and 10 to the negative 5th for the concentration of hydroxide ions. And remember that even though 9 seems like it's larger than 5, it's 10 to the negative 9th, which is actually less than 10 to the negative 5th, right? Because 10 to the negative 9th has more zeros after the decimal point, which means that the hydroxide ion concentration is greater than the hydrogen ion concentration, which means that it is a base. So now let's talk about buffers. Changes in pH can be very harmful to an organism. For example, the pH of human blood must always stay close to 7.4. And buffers will minimize the changes in pH by maintaining the concentration of hydrogen ions and hydroxide ions. So it does this by absorbing hydrogen ions when there are too many and donating hydrogen ions when there are too few. So for example, we have carbonic acid in blood. So let's look at that. So as you can see, the arrow going from left to right, when there's a rise in pH, carbonic acid will become a hydrogen acceptor and a hydrogen ion. So it's basically releasing hydrogen ions into the blood to decrease pH, because again, a rise in pH means that there's a low amount of hydrogen ions. So the donor will donate some hydrogen ions so that the pH will go down. And if we look at the arrow going the opposite direction, if there's a drop in pH, hydrogen ions will be accepted by the hydrogen ion acceptor, and hydrogen ions will basically be taken out of the blood, right? Because a drop in pH means it's getting too acidic. So hydrogen ions must be taken out to raise the pH level to normal. So buffers are really important. Now let's look at isomers. Isomers are compounds that have the same molecular formulas but have different structures and properties. So basically, an example would be glucose and fructose. They both have the formula C6H12O6, but they're not the same. They're structurally different. So there's 3 types of isomers that we're going to talk about, starting with structural isomers, which differ in the arrangement of their atoms. So here's an example. As you can see, all 3 molecules have the same amount of hydrogens and 1 oxygen, but the placement of that differs, which changes the compound and its properties. Next, we have geometric isomers, which differ in arrangement around a double bond. So here's an example. As you can see, again, same amount of every element, but the positioning of the side groups is different, which changes the compound. And last, we have enantiomers, which are mirror images of one another. So think about like your right and left hands. They're mirror images of each other, but they're not the same. So here's an example. And this mirror image, they're not the same. They have different properties and will react differently. So now let's talk about functional groups really quickly. These are very commonly involved in chemical reactions, and there are some important ones that you should know, including hydroxyl, which is oxygen connected to a hydrogen, and then R just stands for like variable group, which will change. But the O and H is the hydroxyl. And then we have carbonyl, which is the C double bond to the O. And again, the two R's just stand for variable groups that will always differ. Carboxyl group is the C double bond O and then the OH, which again, we should remember is the hydroxyl group, but that's connected to the C and the double bond O. And again, R is the variable region that will change. Then we have the amino group, which is the N, H2. And again, R group, variable group. And then we have sulfohydryl, which is the sulfur connected to hydrogen, and then a R variable group. And then we have phosphate, which is P, and then 4 O's, one of which is a double bond. And that's it for this video. Thank you so much for watching, and please subscribe if you'd like to see more videos.